drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today. Introduce yourselves and talk about what you guys study.
Sure. I'm Tim Yeatman. I'm a surgical oncologist by training. I'm currently a professor of surgery at the University of South Florida, and I'm an associate center director for translational research and clinical innovation at the Tampa General Cancer Institute, Tampa General Hospital Cancer Institute.
I'm Ganesh Halade. I'm associate professor in the Department of Internal Medicine, a division of cardiovascular disease. And my research is more focused on inflammation and resolution signaling that focuses on the chronic inflammation that intersects not only cardiovascular disease, but also the cancer and other lifestyle related disease.
Mm-hmm. And you guys have a really interesting new paper out. I read it just because it's the type of thing that comes into my my alert box. I've been talking to a lot of people on the podcast and reading on my own about topics that connect things like inflammation and diet to various chronic disease states, including cancer. And we're going to talk about lipids and diet stuff. We're going to talk about inflammation, and we're going to dive into your new paper. But there's also some background I want to get into at different levels. I want to give people a sense of how you guys think about chronic inflammation and factors that feed into it and cause it, including diet, including dietary lipids in particular. I want to talk about cancer and how you guys think about it and how the field has thought about it historically. And one place I thought was interesting to start, because I'm certainly not a historian, but I do love the history of science. You mentioned at the beginning of the new paper a man named Virchow. So who was this, and what were his ideas about what cancer actually is?
So maybe I can address that. I mean, Rudolf Virchow was I think he was born in 1821 and lived to 1902. He was a pioneering German physician and pathologist, and he's often regarded as the father of modern pathology. Um, and basically, he introduced the concept of cellular pathology, where he looked under the microscope at various tissues and tumors, and it's been attributed to him that he saw first inflammation in cancer, and inflammation was the influx of white cells basically into a tumor, immune white cells. And I think it's been also attributed to him and others that the concept of cancer is like a wound that won't heal. And I always you remember you've had animals like a dog where they have sunspots, and that wound just won't heal. They keep licking, won't heal. It's sort of an ulcerated wound that won't heal. In a human, it might be a diabetic ulcer that won't heal, or an infected wound. And so he saw that, and remarkably, in the 1800s, identified the inflammatory process as being a component of cancer.
Hmm.
Yes. So the cancers, the Virchow actually started the concept like how the inflammation is a fundamental there in context of developing towards kind of continuous process and uncontrolled. In reality, if we see the inflammation is designed helpful, but when it is in the acute process, and that is coordinated by the 1% of the immune cells that we are kind of having since kind of long time of history of the human beings, and that's kind of real historical perspective that how our immune system is built. And in that one, the Virchow added the kind of real key that how this chronic inflammation is involved, not only the acute inflammation, not only the kind of various type of metabolic diseases, but also in the cancer and many other diseases.
Mm-hmm, mm-hmm. So is an idea here that inflammation, sort of on the one hand, we want inflammation, we want our bodies to be capable of this. It's a part of clearing out infections, it's a part of clearing out debris and tissue debris when we get physical damage. But I think a key thing, at least for me and my understanding of inflammation, is you want it to be very spatio-temporally regulated. You want the inflammation to turn on at the appropriate time and then turn off at the appropriate time. You want it to be at the site of an infection or at the site of damage, but not anywhere else. You don't want it to bleed out to other tissues, because there's always this chance of collateral damage. And I don't know much about cancer biology in particular, or the history here, but it sounds like this idea that maybe goes back to Virchow is that if the inflammatory process itself becomes dysregulated, that can actually be a factor in causing cancer.
Yeah, so let's just go back a step. So evolutionarily, you might say that inflammation evolved as an important component of human existence and mammalian existence,
Mm-hmm.
because the greatest threat to life early on was probably infection,
Mm-hmm.
cuts and viral infections and bacterial infections. So we evolved to try to fight those things off. So inflammation, the whole body is geared towards turning on inflammation for that reason. I think evolution,
Mm-hmm.
it just kind of makes sense, right? That
Yeah.
you'd really want to be ready to attack anything that comes at you. But you're right. If that inflammation persists inadvertently, can it cause problems? And I'll give you some great examples in cancer. So it's well known that inflammatory bowel disease, and that's ulcerative colitis and possibly Crohn's, but ulcerative colitis is a great example. In that disease, you see what's called leaky gut. And I
Mm-hmm.
always thought leaky gut was kind of a funky term that no one really understood. But the truth be told, it refers to the epithelial cells that normally have tight junctions, like your fingers sticking together, and they open up. And when they open up, they can release basically blood cells, lymphocytes, blood cells, neutrophils into the bowel lumen, which is the stool.
Mm.
So it's interesting that the most effective diagnostic test for active UC is something called calprotectin, stool calprotectin. Calprotectin is a neutrophil specific marker. So if
Mm.
you see that in the stool, that means you've got neutrophils leaking from your capillaries into the stool, and that's why
Yeah,
people with ulcerative colitis have bloody diarrhea.
I
So
see.
they now you understand that's what true leaky gut is, right?
Hmm.
And when you leak that gut, then you can have bacteria going both ways. And we should get a discussion of microbiome in the future, but let's hold that put on pause for right now.
Hmm.
So you get this leaky gut and this inflammation, and it doesn't seem to go away much. Although in ulcerative colitis, it's called relapsing and remitting
Yeah.
because it's sort of, you get inflammation, then it sort of heals. You get inflammation, it sort of heals. So there may actually be an attempt at resolution of inflammation in that disease. But if it goes on for long enough, inflammation, then you can get cancer. So it's well known that in ulcerative colitis, let go for 10 years, you increase your risk for cancer. That's why people with ulcerative colitis have to undergo uh, surveillance colonoscopy, and they get biopsies every 10 centimeters throughout their colon when they have this long-lasting inflammation, because there
Hmm.
is a risk. The other disease would be Barrett's esophagitis, a risk for esophageal cancer, some skin ulcers, cancer. So it's just sort of this chronic ulcerative condition that can lead to cancer. And ulceration, by the way, is a bad sign for all cancers. If you take melanoma, an ulcerated melanoma has a worse prognosis than a non-ulcerated one.
Hmm.
And why? Well, one theory I've got, and it's interesting. We've always thought of cancer as a true genetic disease, and it
Right.
is, right?
Right.
And from Bert Vogelstein and his group identifying APC, as a sort of the initial seminal mutation in many of these cancers, and subsequently getting KRAS and P53 mutations and other things like that. Um, we now know that maybe metabolism is part of this too. So
Hmm.
could cancer be a metabolic disorder as well as a genetic disorder? That's my question, and I think the answer is yes. So we now know that inflammation appears to be present in the tumor microenvironment, and that inflammation likely causes immunosuppression and reduced immunosurveillance. So
Mm.
one possibility would be that when you develop these common mutations in your billions of cells lining the colon wall, and that happens every day, and
Hmm.
they're normally destroyed by
Yeah.
your immune system. They become apoptotic and they go away. But what if you have an immunosuppression locally, a chronic immunosuppression? Would more of those take hold or take foot? And one example of that is a mouse study, and there's been a number of them, but there's a mouse model called the APC min mouse. And
Hmm.
in that mouse model, they basically have a single hit. In APC can have two hits, but one hit in the APC allele, and the mouse is born with that in every cell of its body. And if you give that mouse a highly obesogenic diet, rich in arachidonic acid, rich in omega-6 effectively, they produce many more polyps, 60% more
Hmm.
polyps than a normal mouse. So you're putting that inflammatory milieu in the presence of a mutation
Yeah.
that could cause cancer. And all of a sudden
Yeah.
you're getting more of them, more
Yeah.
hits.
Double
To
whammy, kind
me,
of.
to me, that's kind of de facto evidence that an inflamed environment could lead to increase. I don't know whether it's increased mutability, but increased chance of mutations taking foot.
Yeah. So if I'm tracking here, because I was actually about to ask you, as someone who's never been a cancer biologist, I never studied cancer biology per se, but as someone who's always in biology, I remember as a student sort of just having the notion in my mind, oh, cancer is from mutation. You get X number of mutations in tumor suppressor genes or whatever, and then the normal cell becomes a cancer cell, and off you go. If I'm following you, Tim, what you're basically saying is yes, there are these mutations involved in creating cancer cells, but normally, I mean, there's so many cells in the body getting so many mutations every single day that all of us at some rate is generating, cancer cells. And oftentimes, more often than not, when things are working appropriately, our body takes care of that. Those cells are detected and they are destroyed, and the immune system is involved in that. It sounds like what you're saying, Tim, is that if you're in a certain inflammatory state, if you have chronic inflammation or what have you, then immune surveillance can actually be sort of turned down such that the probability that any one of those cancer cells that would have very likely been destroyed before it became full-blown cancer and problematic actually doesn't get destroyed. And so even though mutations are necessary to generate a cancer cell, the inflammatory state of the tissue can also dictate sort of the probability of whether or not that cancer cell will actually be handled when it should be.
Right, and we call that tumorigenesis, by the way. So, or carcinogenesis or tumorigenesis, basically the increased ability of the metabolic state to enhance tumorigenesis is what we think could be happening.
Mm-hmm.
It's also possible that once you have a tumor, that the inflammation can make things worse.
Mm.
And we don't know whether there's a threshold of inflammation or whether, in other words, you just exceed this threshold of inflammation, it's all bad, or whether
Yeah.
there's a sort of a linear or logarithmic relationship between inflammation and immunosuppression.
Yeah.
We do know that many of the cells in the tumor environment are macrophages, I think about 50%. And many of those are immunosuppressive macrophages.
Hmm.
And we know that the card-carrying currency of the TME is lipid mediators. And so having a lot of these inflammatory lipid mediators in the tumor microenvironment likely contributes to this immunosuppression and decreased immunosurveillance.
Mm-hmm. And when you say lipid mediators, is the basic idea here that you've got all of these different immune cells. Some of them can stimulate inflammation, some of them can suppress the immune system. When you say lipid mediators, you mean that these are molecules that immune cells have or they release that are sort of the information signals that are saying turn inflammation up or down and things like that?
I'm going to let Dr. Halade, the expert on, take you maybe through
Yeah.
the pathway from maybe a seed oil that has linoleic acid,
Mm.
to arachidonic acid, to how you get to this
Yeah. Yeah.
these lipid mediators and what they actually are.
Yeah.
Sure. Yeah, I like to kind of glad. Before that, I just want to use the analogy for the chronic inflammation, like, what happened when you have acute inflammation. It's just like, when there is a fire, firefighters come
Mm-hmm.
very precisely on time, control the fire, and they leave. But in the chronic inflammation, the first thing is these firefighters don't come on time. And
Hmm.
even if they come on, they just hang on where there is a fire and they forget to leave. So the immune cells get in signal, their transit signal and get out signal is totally discoordinated. And that's where the Tim Yeatman's really nicely kind of explain how the immune suppression is developing. So that's the discoordination of that leukocytes that's developing the chronic inflammation. That
Hmm.
is kind of where many metabolic diseases are kind of developing the whole process.
I see.
Now
So,
we have to get
you have signals calling in the firefighters, separate signals telling them when to leave, and if these things aren't properly coordinated, the firefighters go to the wrong place at the wrong time.
a wrong time, exactly. And plus, especially if they are coming late at the tumor site, plus just they are hanging, doing the party with the fats that every day we are taking in the body, and then just there is a continuous progression of the tumor, and that's whole tumor environment we have described in the paper. That's now here is the question of how these molecules are formed. And we know that historically that these fatty acids, they are not synthesized in our body. That's why they called as essential fatty acids. And then those are necessary 1 to 3 to 5%, depending on where we are on the growth curve in our life. Like if we are like early in the adult phase, we need slightly kind of more quantity. And then down the line, as I'm old or, in the kind of late phase life, we may need relatively lower. But what happened to these fats or essential fatty acids? They get used by the immune cells more rapidly in response to acute inflammation and make number of lipid mediators. Same process happen in the chronic inflammation where these fatty acids used by those discoordinated firefighters or the immune cells where these fatty acids from the diet get converted into the second generation of those molecules. And those second generation molecules we call as lipid mediators or the bioactive lipids. And here we need to note this point that these second generation molecules are more potent than the actual original fatty acids present in the diet. And as you know, and that the standard American diet is enriched with the linoleic acid, that linoleic acid get converted into arachidonic acid, and that further forms the many second generation molecules, and those are the inviting the immune cells. That is the first step of the initiation of the chronic inflammation. So that's how these lipid mediators forms a continuous process that invites more and more immune cells that leads to the tumor progression and kind of continuous growth of the tumor in various regions of body depending on where it is initiating. That is one step. Now, in contrast to that, if you think about, imagine instead of this unhealthy fat, you have the healthy fat, which is coming from omega-3 and other kind of similar related sources, fish or other related sources. And then the same enzymatic system and the immune cell converts those to the healthy molecules. Those are called as a resolution mediators, and that helps to control the immune cells or give the immune cells to get out signals, and there is a clearance of the system and limit the inflammation. So there are totally two arms of the process in the body. Imagine if we have unhealthy fat, then our enzyme system with the help of immune cells, then that unhealthy continuously kind of feeds the chronic inflammation. In contrast, if we have a healthy fat, then our immune system with the help of this immune responsive metabolic enzymes make the molecules, helps to clear the system. So that's how this process, if it is really balanced, then we stay healthy, but if it is imbalanced, then leads to continuous chronic inflammation. But there is a very tight coordination of the fat, what we take every day, and the enzymes present in the immune cells, especially the leukocytes, that determines the biosynthesis of those lipid mediators. And down the line, those lipid mediators, those are very potent than the fatty acids, actual fat in the diet, and gives the clear signal to our immune cells.
Mm-hmm. So we've got these lipid mediators that are super important for the inflammatory response, how it's going to initiate, how it's going to resolve. And in essence, it seems like there needs to be a balance of sort of two classes here, the pro-inflammation mediators and then the resolving mediators. And these have to do with things like dietary fatty acids. So we eat fat. Certain fatty acids serve as precursors to these different lipid mediators. One thing I want to talk about here is this concept of balance versus imbalance that's going to become important, I think. So is it true that if I just eat a bunch of fat, let's just say I'm not tracking it too closely. That's probably true for a lot of people. I'm not paying too much attention to the specific fatty acids I'm eating. Seed oils, saturated fat, omega-3s, you name it. I'm just sort of eating what I eat. Can't my body doesn't it just know how to make the perfect needed balance of these things? Or does the balance of what's coming into me from my diet really dictate whether or not there's going to be an imbalance?
So have you ever heard that phrase, you are what you eat?
Yeah.
I think there's some truth to that. So we have to, again, get into the biology of this a bit, but the enzymes that process omega-6 derived fatty acids or omega-3, they're actually the same. The ones that process omega-3 and 6 are the same enzymes. So it's called ALOX5 and 5-AP. And those enzymes can handle either one, the pro-inflammatory or the pro-resolving lipid precursor, omega-6 or omega-3. So as with any enzyme, if you give it a substrate in excess, it's going to favor that substrate if it's got equal propensity to to affect both.
I
And
see.
so if you load your body up with omega-6s at a ratio of 30 to 1 or 25 to 1, your enzyme is going to be more likely to convert that to a pro-inflammatory lipid mediator than a pro-resolving lipid mediator. And that's kind of, we think, what happens where we have, our diet is so overwhelmed with omega-6s, you can't eat enough omega-3s to balance it. The lipid mediators are evanescent, by the way. They get turned on and off, like you said. It's a temporal thing. It's timed with inflammation. And there's a certain time when these things come on. There's a certain time when they should go off. But the substrates are always floating around, right? And so there's a nuance to the science here, understanding what the substrates are and then what the lipid mediators are and measuring both. So if you look at it, and I looked at myself, I have a 13 to 1 ratio, for example. Ideally, it'd be 1 to 1 or 2 to 1, favoring, more towards the omega-3. You'd like two omega-6 to 1 omega-3.
Yeah.
But like I'm 13 to 1, but the average American may be 25 to 1 or 20 to 1. Now, if you look at the foods we eat, let's take beef, for example. Beef or pork or anything of that nature, it's about 25 to 30 to 1 because the beef is grain fed. And you've heard of fattening the calf with grain, using grain to fat. Well, that's why it's basically this sort of carbohydrate rich diet and there may be, by the way, maybe soybean oil in things like cattle feed. I don't know for a fact. But if you're getting a grain fed animal, that's where the meat is now 25 to 1. And you're getting every bread that you eat, or every cookie or piece of pie or whatever you're eating is loaded with, basically omega-6s, then your body's going to see much more of that. So you are what you eat. I think you're going to be flooding your receptors and your enzymes with this inappropriate ratio.
Yeah.
Now, what can you do to correct that? You can, well, like if you go to grass-fed beef, the ratio is 1 to 1.
Yeah.
Amazingly. And grass-fed milk, 1 to 1. And grass-fed butter, 1 to 1. So just switching to grass-fed, animals and milk can really help, because many people eat a lot of meat and butter and milk. And so that's one thing you can do. The other thing you can do is try to be vigilant, looking at labels and seeing what you're actually eating. But you have to have a microscope to get the labels. When you see so many ingredients, it's hard to actually find where the soybean oil is, but you can see it if you look very carefully.
Yeah. Well, I mean, I think one of the places, one of the things implied here is, it's not sufficient just to look at the total fat content. You've got to actually go down at least one more level and say how much is polyunsaturated, how much is saturated, and so forth.
There's a lot of literature. We grew up, I would say, in the early days where it was butter and Crisco. My grandmother was known to have made a jingle for Crisco: north, south, east, west, creamy Crisco is the best. And of course, we got away from all that because we realized that saturated fats were bad too in excess, right? And they can cause heart disease. And then I think when people moved over to margarine and things like that, maybe that helped heart disease to some degree. But when you go full spectrum and put every food you have loaded with these types of oils, then you've changed the balance. So you may have reduced the saturated fats, which is good, but you've increased the polyunsaturated to the inflammatory side, which can be bad. So again, life is a balance, and eat in moderation is the right approach. Do everything in moderation.
Yeah. And so I want to get into the idea of balance more and really dig into some of the biochemistry before getting to the paper. But just to tie off on some terminology, because we're going to have people listening with variable levels of knowledge coming into this. So when you talk about, I want to sort of make it clear for people, what are omega-3s? What are omega-6s? When you guys talk about omega-6s, you're talking about certain polyunsaturated fatty acids that come to us from our diet. And Tim, a number of the things that you said, and I'll let you guys expand on this, but a number of the things that you said are the average American, the average Westerner, probably the average person on planet Earth at this point, is getting an imbalance of omega 6s and omega 3s. So we're getting way more omega 6s, most of us, compared to omega 3s. Historically, in evolutionary terms, if we go back before industrialization, back in time, most people were consuming a pretty balanced ratio of these omega 3s and omega 6s. And these omega 6s are what people refer to as either vegetable oils or seed oils. Is that correct?
Yes,
Yes.
that's correct.
That's correct. So what happened is kind of now our source of the kind of food intake is slightly changed. We moved from home cooked food to the processed and packaged food. So that's the source. Then from there, why the processed food industry will use the healthy oil for your health. So the majority they use the seed oils, which are much more cheaper, so that they can make more revenue, but that is costing to our health because of the presence. Now, for that one, we will use the easiest way, the car analogy. So what happens if our car oil goes bad? We go and take out the old oil and put the new oil. But here in the human, we don't have that knob to take out the old oil. And even if we go switch to another healthy, but their old oil is still there a long time in the body. This is very simplified version of this. And for the work the car perfect manner, need a kind of sort of four wheels. Same way happens into the fat. So the fat is the number one component that we take from the environment, from different products. Then that get used by the enzymes which is present our in body. And then the third product is the lipid mediators. And once these lipid mediators are formed, then the last wheel is the receptor. So the diet, enzymes, lipid mediator, and receptors, those all are the four wheels that makes or decides whether the fat is going to either helpful or harmful side. Now, the first step itself, our body is fully loaded with the omega-6 continuously from the environment, then obviously it's driving towards the pro-inflammatory side. Obviously, it has no choice that the enzymes using that because of the abundance, making more pro-inflammatory molecules, and the receptor related to the pro-inflammatory molecules, those are constantly activated. So the body is kind of going towards the pro-inflammatory. This is kind of super simplified version of very complex pathway, because one single fatty acids like omega-3 linoleic acid gets to the arachidonic acid. The arachidonic acid makes more than 20 to 25 molecules at the site of tumor or at the site of injury, depending on the progression. So that's how the fat biology, even it's very complex, but this is kind of very simplified version that how the imbalance of the omega-6 to omega-3 is kind of leads to that point of the chronic inflammation, because of these key four components driving or feeding the chronic inflammation.
Yeah.
So
So, oh, go ahead, Tim.
I was going to add to that you can look at omega-6 to omega-3 ratios in body fat. And over the years, from 1950 to more recently, there's been a tremendous rise in the omega-6 to omega-3 ratio in the Western population. And so that's, prima facie evidence that you're taking more in and the diets have changed.
Yeah.
And so that's a fact. And the other thing that's interesting is fat, when you eat fat, it can go everywhere in your body, basically. So there's a guy named Garth Nicholson who identified the lipid bilayer of the cell membrane is, two lipid bilayers, right? So, or two layers, a lipid bilayer. And phospholipids, right, compose this the cell wall of all our cells. So those fats that you're eating get inserted into every cell in your body.
Yeah.
And so over a course of decades of eating, the wrong diet, you can imagine that all your cell walls have that makeup, right? That constituency. The other thing that's interesting is there is a enzyme called phospholipase A2. And that's an enzyme that releases arachidonic acid from the lipid cell wall. And phospholipase A2 is actually a cardiac test for inflammation that you can actually get with LabCorp and Quest. You can measure your level of inflammation, and I think it's different than measuring C-reactive protein that a lot of people use.
Mm-hmm.
So
Mm-hmm.
it's interesting. How do you determine inflammation in the body?
Right.
One is to look at phospholipase A2
Yeah.
levels, and
Yeah.
they're active in releasing arachidonic
Yeah.
acid.
And
So.
Tim, I'm gonna say here that I want to come back to that maybe because one of the things that often gets brought up in this realm among aficionados or people who claim to be is there's some maybe gaps in the clinical literature where people will look for things like CRP or certain inflammatory markers, and they'll say, well, we don't really see a correlation between intake of these things and inflammation. Inflammation meaning like the one marker
Right.
they look for. I think what
Right.
you're saying is you have to look for the right markers in the right places in order to pick things up properly.
Absolutely. And I think that, plasma levels of things are different than tissue levels of things.
Mm-hmm.
So people will look at plasma levels and say, well, I didn't see anything there. But you look at the tissue, it's a different story. So I think the science is still in its infancy in understanding lipidomics and lipids
Yeah.
we eat. And it's remember, people are complex. And
Right.
you also remember there's heterogeneity. So if you look at, one person, another person, even our own data, we saw some people with sort of very high LTB4 levels and some people with less high and some with lower levels. And you say, well, on par, they were all elevated. But why is the heterogeneity there? What does that mean? And
Yeah.
again,
Yeah.
I don't know yet. We're going to look into it with more samples. It's certainly possible there's a threshold that you exceed. Like I said before, where if you exceed a certain threshold of like LTB4, you're inflamed.
Mm-hmm.
Or it may be that a really high level is worse than a really low level.
Yeah.
We don't know yet, but clearly there's an on par average median elevation of these
Yep.
things.
But okay, so let me see if I can tie some things together and maybe simplify a little bit and then dig into what if I'm hearing you guys correctly, some of the key biochemistry for why certain things are true. So you've got omega 6s, you've got omega 3s, you've got other types of fatty acids, but if we're just talking about the PUFAs, the polyunsaturated fatty acids, two big classes, omega 6s, those are what we refer to when we're talking about seed oils. Then you've got omega 3s, that's typically referred to as fish oil. Omega 6s are going to serve as the precursors to the pro-inflammatory lipid mediators most of the time. The omega 3s are basically going to serve as the precursors to the resolving lipid mediators. And so you want these things in balance. And it sounds like it really does matter what your balance is on the intake side, your fatty acid intake, the food you're eating and its fat content, because when these things are the reason these things go out of balance, I think ties to what you said, Tim, which is they have the same enzyme is responsible for processing both of them. And so there's sort of like this rate limiting thing or this funnel where if you've got an imbalance on the intake side, then you're going to produce more of the one class of lipid mediators on the output side of that enzyme because it's the same enzyme processing both the omega 6s and the omega 3s. And so you're unfortunately your body, it would be great if your body could just take an arbitrary set of fatty acids and then make exactly everything in perfect balance. But based on this biochemical limit here and how these enzymes work, that's not the case. And so your intake really needs to be balanced, if I'm hearing you guys correctly.
And supporting that, there are some studies that are very interesting. We insurance companies have used, LDL levels and say, we get your LDL level below 100 and that's healthy.
Yeah.
Get your HDL level up a little bit, that's healthy. But I don't know whether anybody's ever used omega 6 to omega 3 ratios. And it might be a better index of health because there are studies that show that a higher omega 6 to omega 3 ratio in plasma is linked to increased mortality risk from all causes. Cancer, cardiovascular disease, individuals have the highest ratios and have had a 26% higher risk of mortality and a 14% higher cancer mortality. So there, and there's some confusion on this, some recent studies where some cancers show a lower incidence with higher ratios, like breast cancer, but it's small. It's like a decreased 8%. So a lot more work needs to be done here, but the concept that omega 6 to omega 3 ratio might be important indicator of health, I think is something that needs to be explored.
Mm-hmm. Mm-hmm. And Go ahead, Ganesh. Were you going to say something?
Yeah, I just want to point out that, as already Tim mentioned, like, you are what you eat, but again, there is slightly more than that there. So when you eat, what you eat and how much you eat, so those are getting granular because down the line, your, everyday biology in context of circadian rhythm contributing to that component. And that's where it is coming to the point that when these lipid mediators form using the processed fat, and sometimes people think that, hey, those are absolutely kind of these seed oils are bad. But one point we just need to understand their name. They called as essential fatty acids, means they are absolutely essential into 1 to 3% for the initiation of inflammation when there is injury or infection, because those molecules are the one gives a signal to our immune cells, hey, there is a problem, come over there.
Yeah.
So those invite the immune cells, that is there.
Yeah.
But here is the what happening is that they are continuously giving that signal, hey, come here at the site of the tumor, here party is going
Yeah.
on.
Ganesh,
So that
I think you said that the omega sixes are necessary at 1 to 3%. What did you mean by that 1 to 3% of calories?
1 to 3% in terms of their, depending on the growth curve. If you are in the adult phase, then probably in the 1 to 3% in terms of the kind of how much you are taking as in the form of the grams, how much you are taking in the diet.
I see.
Yeah,
So
yeah.
grams. So a tiny percentage should be.
Yeah, tiny
Yeah.
percentage, tiny percent, and that also needs to kind of club with how much you are active versus what's your sleeping pattern. So
Yes.
those all three components necessary for the activity of the enzymes machinery. What the team was mentioning, that because this machinery process both the omega-3 and omega-6 in response to injury,
Hmm.
or the kind of tumor where there is a continuous chronic inflammation, where the lipid mediators are formed, that we have quantitated by using very state-of-the-art mass spectrometry.
Yeah. But it's go ahead, Tim.
But I wanted to get back to, what's healthy and what's not healthy. So, we think a balanced ratio of omega-6 to omega-3 in your plasma, a balanced ratio in your food, therefore, makes the most sense. Close to 1 to 1 as possible. And again, the UK Biobank study of 86,000 patients found increases in all-cause mortality and cancer mortality and cardiovascular disease increased with the increased omega-6 to 3 ratio. So that's really good evidence. But if you go the opposite direction, let's say you really remember, omega-6s are essential fatty acids. You have to have them to live.
Yeah.
But it's the question of how much, right? But if
Hmm.
you go the opposite direction and try to reduce your ratio from 1 to 1 to 1 to 4, in other words, 4 on the omega 3 side and 1 on the omega-6 side, that's very hard to do. But apparently the Inuits of Alaska have that sort of ratio, and they have a high risk of heart disease. So if you
Mm.
imbalance it the other way, you may actually increase problems too. So it's this balance, this life balance that Dr. Halade often talks about,
Yeah.
is important.
Yeah. And so getting to the question, so we talked about sort of the biochemical reason why balance makes sense and 1 to 1 makes sense, because the omega 6s, omega 3s kind of get processed by the same enzyme. And in some sense, you might say that the body is expecting balanced input. And so I want to ask if that's true. And so if we think about this from an evolutionary perspective here, is would you say it's accurate or reasonable to say that for most of human history, before we started industrialization, when we were, out in the wild, so to speak, except in extreme environments, perhaps like where the Inuit lived, Tim, that in most natural environments, humans were we were intaking a 1 to 1 ratio of omega 6s to omega 3s because that was what was more or less naturally present in the environment. It was only after we started creating synthetic environments, industrial environments, or, in maybe extreme cases where some people moved up to the Arctic, where you got this imbalance of omega 6 and omega 3 inputs.
Yeah, you're absolutely right. So modern agriculture, big agriculture, has tried to mass produce foods at a lower cost.
Mm-hmm.
And so you're right. In those days, I'm sure that all beef was grown on grass. and had this 1-to-1 ratio. And I hear that it takes about 3 years to bring a steer to market on grass-fed
Mm,
versus maybe 1 year on grain-fed. So there's an economic advantage to go with the grain feeding,
yeah.
and that's where we've gone. But the same thing's true with these other oils. It's a lot cheaper to put in canola oil, which is horrendous processing to get there from seed. If you actually read and look how it's actually processed, or soybean oil or cottonseed oil or sunflower oil versus an olive oil or avocado oil. Those are more expensive. So we've put these cheaper oils into things and tried to promote the idea that omega-6s are healthy, but in fact, just this imbalance has it's in everything now. So you can't go overboard, and we have in all our foodstuffs.
Yeah. Yeah.
Yeah. One thing happened is that our kind of food source is changed, and that is shifting the omega-6 to omega-3. But at the same time, what happened? We have really kind of our whole lifestyle becomes more sedentary. So
Right.
we increase our pro-inflammatory input in the body, but same time we become sedentary. Another one confounding factor is added, that we are less sleeping. So that kind of what we may ask, like, okay, what is the connection of these two components? One, that is kind of because of the pro-inflammatory input, the body is kind of moving towards the obesity, and that kind of correlate to the kind of number of high cases of the cancer. And the component, the lesser, these both the components actually impacting the enzymes which processes the fat, whether it is omega-3 or omega-6. Then
Hmm.
if your enzymes become weak because either overabundance or your sedentary lifestyle or you are not sleeping on time, that leads to obviously what happened, that whatever intake you are taking, your machinery to process the fat is already cramped, weak. So the quality of mediators will be a very fragile or totally collapse or disrupt. And that's why whatever you are eating, it's not getting cleared, or whatever the debris is from different sites in the body,
Hmm.
that is not clear. That is the kind of primary function of the immune cells when you are sleeping, as a patrolling different immune cells, go in a different sites, find out the wear and tear and clear it. So that's how kind of hold the immune system and the fat, they have very tight coordination every single day, and find out where is the debris, how it can be cleared with the help of those four components: the fat, enzymes, lipid mediators, and the receptors. And they work as a very precise, tight fashion so that they can clean the body and maintain the health.
Yeah. And then so one of the themes here I think is that there are multiple points of variability here, or where the potential for inflammation can be altered. So one is just what's the total amount of fatty acid substrate you have to work with to begin with, and that gets into this idea of the omega-6 omega-3 balance in terms of our fat, the fat component of our diet. But also, Ganesh, as you're saying, there are other factors that determine the extent to which true inflammation will come from that potential, and that ties into things like lifestyle. How much are you moving? How sedentary are you? How much stress is there? What are your sleep patterns like? All of that will tune the extent to which that inflammatory potential from the omega-6s actually then gets turned into bona fide inflammation in the body.
Nick, you're describing systems biology. So systems biology is the concept that we're not you just can't look at one molecule, one cell in isolation. You have to understand how everything is orchestrated as a system, and that when you perturb the system, it's like the butterfly effect. Sometimes you can perturb many different things, and I've always thought that genetic engineering was interesting, right? I actually wrote a paper years ago, Egypt in the year 2000. And this was a paper written in 76 predicting the use of genetic engineering in agriculture. But the reality is that when we put a single gene into a cell, do we really know what effect that one protein produced by that gene or knocking out a gene is doing on the other 20,000 genes and 150,000 proteins? Are there subtle changes in patterns we don't know that can stimulate autoimmunity in the body, for example, and all of a sudden your body's recognizing a form of wheat that is not natural in the sense that there's a genetic
full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. Hey everyone, I want to share with you a new AI-powered research tool I've been using to find scientific papers and learn from the literature. It's called Consensus. It is an AI-powered search tool built using large language models trained to help you find and understand the best science faster. Consensus helps you not only find individual papers, but its AI models will help you assess research quality and even synthesize insights across multiple papers for you. You can ask it general questions about any topic you might have, such as what are the known risks and benefits of a certain prescription drug? What do we know about how a certain food or nutrient affects the human body? Anything you can think of. Its Pro Analysis feature will actually provide you with a concise, written summary of what the literature says, broken down point by point, highlighting the strength of the evidence across papers, and providing you with individual studies ranked by the relevance and including additional information like citation rates, journal quality, and more. You can also save, cite, and share individual studies with a single click. Unlike other AI and search tools like ChatGPT, PubMed, or Google Scholar, Consensus uses over 20 custom large language models that are developed and trained for this specific purpose. The base Consensus product is totally free, but the premium product that you can subscribe to has better features and unlimited queries. And I highly recommend this if you are using outdated tools like PubMed or Google Scholar, or you just do your own research. For a limited time, you can use the code MINDMATTERSPECIAL, all one word, to get a free premium subscription for 1 year. That is a $150 value for free if you use the code MINDMATTERSPECIAL today. So click the link in the episode description or check my support page and give Consensus a try. Use the code MINDMATTERSPECIAL to unlock your free premium subscription today. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.